Alright, the photosynthesis is a process that autotrophs perform in order to use energy contained in the photons of sunlight to produce organic compounds that are necessary for for survival. And this process will occur in the chloroplast, and there's two main parts to it, which is the light-dependent reaction, and they take place in the thylakoid membrane of the chloroplast, while the Calvin cycle will take place in the stroma of the chloroplast. The light-dependent reaction, H2O and photons are consumed, which then NADPH is produced by reducing ATP to move to the Calvin cycle in the stroma. In the Calvin cycle, carbon dioxide and the ATP and NADPH from the photosystems are consumed, and CO2 is reduced into an organic result of the Calvin cycle. ADP and phosphate and NADP are released, which are sent back to the light-dependent reaction. Moving on to the light-dependent reaction, this process will occur in the thylakoid membrane of the chloroplast, and these inputs are from photons from the sun, H2O, and NADP+, as well as ADP and phosphate from the Calvin cycle. Chlorophyll molecules will absorb photons, and the energy of the photons is used to move electrons up to a higher energy level in the photosystems too. And electrons are then sent through the electron transport chain in the thylakoid membrane. From there, H2O is split up by photolysis, and its electrons are used to replace electrons in photosystem 2, which are then sent to the electron chain and released as oxygen as a side product. The electrons in the chain will then go from high to low energy as they move through the transport chain. The energy will be used to pump hydrogen ions from the stroma into the thylakoid to establish a type of protein gradient. These proteins are then moved through ATP synthase as they move from high to low concentration and back out into the stroma. From there, phosphorylation ADP to make ATP will move through the process of chemiosmosis, and the ATP is sent to the Calvin cycle. When the electrons move to photosystems 1, they are then transferred to chlorophyll molecules, where they are once more energized by photons. These high energy electrons will go through another electron chain, and in the end they are transferred to NADP reductase along with NADP+, and NADP+ is then reduced into NADPH, which is an electron carrier that is sent to the Calvin cycle. These electrons that photosystems 1 loses are replaced by the electrons that are in photosystem 2. Alright, now moving on into the Calvin cycle. This cycle occurs in the stroma of the chloroplast, and there's 3 main stages: carbon fixation, reduction, and RUBP regeneration. For the cycle, the inputs are carbon dioxide that the plant takes in through its stomata, and the ATP and NADPH made in the light-dependent reaction. Carbon dioxide provides all of the carbon and oxygen that are required to make glucose, the main end product of the cycle. NADPH is an electron carrier which carries electrons from the light-dependent reaction to the Calvin cycle. And it's significant for the upcoming steps, starting with the carbon fixation, where 3 carbon dioxide are joined with 3 RUBP with the help of the enzyme Rubisco. And this carbon fixation then creates a 3,6 carbon intermediates, which is split into 6 carbon molecules called 3-PGA. Next, 6 ATP is used to turn 6 3-PGAs into 6 BPGs by adding phosphate to the 3-PGA, and ADP is released. Next, NADPH is oxidized and reduced to 6 BPGs, which then are turned into 6 GRPs, and NADP+ is released. 1 GRP then is taken off the cycle, and the other 5 are sent to the last stage, which is RUBP regeneration. This carbon and the 5 GRPs are rearranged and ATP gives off its phosphate groups to make 3-RUBP, which are used to repeat the cycle. The ADP and NAPDH that the cycle uses are sent back to the light-dependent reaction and get turned into ATP and NADPH. And then the G3RP that the cycle forms can be turned into glucose or other organic and plant needs.
C3 photosynthesis is the basic unaltered version of photosynthesis, where there are no specific adaptations to photorespiration, and this is used by 85% of plants. In C3 photosynthesis, the Calvin cycle occurs in its base form without any alterations. So starting out, 3 CO2 are fixed into 3 RUBP via Rubisco, and this forms 3 unstable intermediate molecules. This 3 unstable intermediate molecules quickly splits into 6 molecules of 3 PGA, and then after that, ATP gives its phosphate groups to the 6 molecules of 3 PGA to form 6 molecules of 3 BPG. And after that, 6 molecules of ADP are released. And then the 6 molecules of 3 BPG are reduced by 6 NADPH to form G3P. And as a result of this, 6 NADP plus and 6 phosphate groups are given off. Then one of the G3P is given off as the result of photosynthesis, and the other 5 G3P are used to regenerate RUBP. And this is done using ATP, which is used to regenerate RUBP. One problem with C3 photosynthesis is photorespiration, which is when instead of CO2, oxygen is fixed by Rubisco. And this forces the cycle to release CO2 and not G3P, which therefore means it wastes energy. An example of a plant that uses C3 photosynthesis is wheat and many other common staple crops. And studies have shown that instead of using C3 photosynthesis, by using C4 photosynthesis, you can get up to 50% productivity gains just through preventing photorespiration. C4 photosynthesis is a version of photosynthesis where the plant has adapted to prevent photorespiration. And photorespiration is when RUBP reacts with oxygen and G3P is not produced, which is bad for the plant. It accomplishes this by fixing carbon beforehand and not during the Calvin cycle by Rubisco. So this prevents Rubisco from accidentally fixing oxygen to RUBP. C4 photosynthesis is physically separated between the mesophyll and the bundle sheath cell of a plant. The process starts in the cytoplasm of the mesophyll. CO2 is fixed to the 3-carbon PEP to form by PEP carboxylase to form 4-carbon oxaloacetate. This 4-carbon oxaloacetate then turns into 4-carbon malate, and then the 4-carbon malate is transported to the bundle sheath cell stroma. Then after that, the CO2 is transferred to the Calvin cycle where it's fixed by Rubisco and it undergoes the Calvin cycle to produce the G3P. And then the malate is turned into pyruvate, which goes back to the mesophyll cytoplasm. After that, ATP gives one of its phosphate groups to the pyruvate to turn it back into PEP. And then this allows the cycle to restart and for more CO2 to be fixed. By doing it this way, it's preventing O2 from ever reaching Rubisco. Now we have CAM photosynthesis. CAM photosynthesis is a process which plants that live in very hot climates use. To intake CO2, the plants have to open their stomata and take in the CO2. But if they do this in hot climates, water will then evaporate out of the stomata through transpiration. To prevent this, CAM plants only open their stomata during the night and store CO2, which they can use for photosynthesis during the day. Therefore, this process is split between the day and the night. During the night, CO2 is taken in from the open stomata of the plant and is transported to the mesothelal cytoplasm, where CO2 is fixed to PEP via PEP carboxylase to form the 4-carbon oxaloacetate. This 4-carbon oxaloacetate then turns into the 4-carbon malate, and then this 4-carbon malate then turns into malic acid, and malic acid is stored during the night so the plant can use it later on during the day for photosynthesis. Then when day comes, the malic acid is turned back into malate, and the malate gives 1 CO2 off, which can be fixed into the Calvin cycle by Rubisco and used to generate G3P. The rest of the malate is turned into PEP, which goes back into the cycle and it stays there until night again, where it can fix more carbon and store it for the day. An example of a plant that uses CAM photosynthesis is pineapples, and this is why they can grow in such arid climates.
Cellular respiration is a multi-step process by which cells convert glucose and oxygen into usable energy in the form of ATP. It begins in the cytosol with glycolysis, where glucose splits into 2 pyruvate molecules, producing a small amount of ATP and NADH. These pyruvate molecules then enter the mitochondrion and are converted into acetyl-CoA, which feeds into the Krebs cycle in the mitochondrial matrix. The cycle generates more NADH, FADH2, ATP, and releases CO2 as a waste product. NADH and FADH2 then carry high electrons to the electron transport chain located in the inner mitochondrial membrane, where chemoosmotic gradient of H+ ions powers ATP synthase to produce a large amount of ATP through oxidation phosphorylation. Oxygen acts as the final electron acceptor, forming water and ensuring the chain keeps flowing. Altogether, this process transforms 1 glucose molecule into carbon dioxide, water, and up to 38 ATP, fueling nearly every activity in the cell.
Glycolysis is a cell's first steps in harvesting energy from glucose, and it takes place in the cytoplasm. The process begins with a single 6-carbon glucose molecule and ends with 2 3-carbon pyruvate molecules. To kickstart the reaction, the cell invests 2 ATP. As glucose splits and rearrange, 4 ATP are produced by substrate-level phosphorylation, giving it a net gain of 2 ATP. At the same time, 2 NAD+ molecules are reduced to 2 NADH, which acts as an energy messenger, carrying high-energy electrons to later stages of cellular respiration. This process does not require oxygen or organelles, and it occurred in primitive prokaryotes very long ago, and occurs in most organisms now, which gives proof that it is a very common evolutionary trait that originated a long time ago, that has been relatively conserved. Once glycolysis is complete, the pyruvate molecules cross both the outer and inner membrane of the mitochondrion and enter the matrix, where pyruvate oxidation takes place. Each pyruvate loses a carboxyl group, releasing carbon dioxide and forming an acetyl group. Then NAD+ captures electrons from the acetyl group, forming NADH. Finally, coenzyme A attaches to each acetyl group, creating 2 molecules of acetyl-CoA ready to enter the Krebs cycle. Alcoholic fermentation and lactic acid fermentation are 2 types of anaerobic respirations that cells use when oxygen isn't available. The purpose of these fermentations is to regenerate NAD+ without the electron transport chain, which needs oxygen. Both begin with glycolysis, where glucose is broken down into 2 pyruvate molecules, producing 2 ATP and 2 NADH. In alcoholic fermentation used by yeast and some bacteria, pyruvate loses carbon dioxide and becomes acetaldehyde, which is an intermediate. NADH then donates its electrons and hydrogens to acetaldehyde, regenerating NAD+, forming ethanol as the final product. This NAD plus can be used to oxidize more glucose to pyruvate, allowing glycolysis to occur again. An example of an organism that uses lactic acid fermentation is yeast. This is why when you ferment yeast, the yeast rises, which is due to the CO2 gas produced by alcoholic fermentation. In contrast, lactic acid fermentation, which occurs in muscle cells and certain bacteria, skips the CO2 step. NADH transfers its electrons and hydrogens directly to pyruvate, regenerating NAD plus and producing lactate. Both pathways allow glycolysis to continue by recycling NAD plus, which can be used to oxidize more glucose to pyruvate, but they result in different end products: ethanol and CO2 in alcoholic fermentation, and lactic acid in lactic acid fermentation. An example of an organism that uses lactic acid fermentation is humans. During exercise, when you can't get enough oxygen, your body does lactic acid fermentation, generating lactate, which causes the pains of exercise. The Krebs cycle, also known as the citric acid cycle, is a key part of cellular respiration that takes place inside of the mitochondrion. After glucose is broken down in the cytoplasm through glycolysis, it produces pyruvate acid, which then enters the mitochondrion and is converted into acetyl-CoA with the help of coenzyme A and NAD+, releasing CO2 and producing NADH. Acetyl-CoA then enters the Krebs cycle, where it spins through a series of reactions that generate high energy molecules. First, a single acetyl-CoA attaches to an oxalidosate to produce citrate. This citrate goes through several changes, and on the way, NAD plus is reduced to NADH. FAD is turned into FADH2. ATP is produced from ADP and phosphate by substrate level phosphorylation, and CO2 is released as products. These energy carriers, NADH and FADH2, move on to the electron transport chain, where they help produce up to 34 ATP. And then after ATP is produced, the molecule undergoes more changes to be turned back into oxalidosate. And on the way, more NAD plus and FAD are turned into NADH and FADH2. For each glucose, 2 acetyl-CoA are produced, which counts for 2 turns of the cycle. In 2 turns, 6 NAD is reduced to 6 NADH, and 2 FAD are reduced to 2 FADH, and 2 ADP are made by substrate level phosphorylation.
The electron transport chain is the final and most energy-rich stage of cellular respiration, taking place along the inner membrane of the mitochondrion. Here, NADH and FADH2, produced during glycolysis in the Krebs cycle, donate high-energy electrons to a series of protein complexes embedded in the membrane. As electrons move through the chain, their energy is used to pump H+ ions across the membrane, creating a concentration gradient. These ions then flow back through a specialized enzyme called ATP synthase, which spins like a turbine to generate ATP from ADP and phosphate. This process, known as oxidative phosphorylation, produces the bulk of the cell's ATP, about 34 molecules per glucose. At the end of the chain, oxygen acts as the final electron acceptor, combining with electrons and H+ ions to form water. Without oxygen, the chain halts, making it essential for efficient energy production.